I'd like to talk to you about the first. Am I being caught here? Okay. I'd like to talk to you about the first, but to give you a brief preview, our work really fell into two large areas. And as you see there, the first, represented in blue, really was about drug delivery. And that work fell into both work on DNA nanostructures and also cell surface targeting. The second was about a bio-oscillator, getting the oscillation system from cyanobacteria, which occurs naturally, into E. coli, where it obviously does not occur naturally. So let's begin with drug delivery. We decided to look at the problem of drug delivery through the lens of synthetic biology. We realized that when it comes to developing a new drug, it's often very expensive and time-consuming to figure out how to actually package that drug and get it somewhere within the body. We felt at the beginning of the summer, and still do, like it would be great if we could have a more modular drug delivery system, one engineered with really just two basic parts. The first part is a universal container into which you can put whatever drug you're developing and want to deliver. And the second part is really about cell surface targeting and about getting that container with your drug in it to a specific cell. And the way we think that this is great is that you can swap in and out whatever drug you feel like. So represented there as a box is our universal container, and you see an antibody being put in it, but that could be any macromolecule or protein. And then we want to bring it to a certain cell, and you see a number of different hypothetical cell types there, and we'd like to be able to swap in and out whatever cell that would be. And that, I think, would greatly improve drug delivery and development. And we realize this is a huge goal for one summer, but we think that we took a great few first steps. And we split up our work in two ways. 4 of us worked on making this universal container, and we were largely inspired by William Shee's work at the Harvard Medical School. And he's been working on DNA and nanotechnology, and we felt it would be really novel and great if we could build this universal structure out of DNA. And we'll get to the details of why we might want to do that in a moment. 2 other people worked on cell surface targeting, and they approached it from 2 different angles, and they'll explain that in a few minutes. Turning to DNA, why would we build a container out of DNA? Well, you might imagine building a container out of proteins, and a lot of people work on protein engineering. But as many of you know, it's an extremely difficult process. It involves a lot of knowledge and a bit of luck, and it's hard to engineer proteins to be just how you want. And that's because you need to know not only the primary structure, but the secondary structure and tertiary structure, and making a modification, it's often hard to predict exactly what will happen. But DNA actually is just the opposite. It's very predictable. And this is sort of all being pioneered right now within the past few years, and as it turns out, as you all know, Watson-Crick base pairing is very predictable. A will pair with T, C will pair with G, and these are very strong bonds. Furthermore, we can make covalent modifications very easily. There are a number of things on the market. You all worked with restriction enzymes and things like that. You can modify whatever you're building out of DNA very easily, and it's cheap to buy and easy to use. Furthermore, synthesizing short strands of DNA is relatively inexpensive. And it turns out that that's all you need to do to actually make surprisingly large structures from DNA. And we'll get to the details of that in a moment. Furthermore, with just a bit of heating and cooling, structures will self-assemble. And all of this leads to DNA being a highly programmable material. And we're used to thinking of DNA as genetic material, but really here we're trying to get to think in another way. Think of DNA as a building material. And you can actually sit down at the computer with very little knowledge of the chemistry of what's happening and just a few algorithmic facts of A pairs with T and C pairs with G, and a few other facts that I'll get to in a moment, and actually determine which short single strands of DNA you need to order to actually make a large structure. Getting to some more details, how does this engineering happen? Well, if we look at the first picture there, you see 2 blue strands and a yellow strand and a green strand. And each of those lines actually represent a single strand of DNA, and we see a crossover between 2 double helices in the middle. Many of you know that as a holiday junction. Really, that's just what it is. And we're actually putting that crossover there by ordering a strand that will bind with the first half of the first blue strand and the first half of the second blue strand. And that's the yellow strand we would actually order. To get to more detail, really this is 3 views of the exact same thing. A more detailed picture is in the middle of exactly how that crossover is happening. And it turns out that if you move where that crossover is down the strand in either direction, you have positional control, and you change where those 2 double helices actually come together. And by having a lot of them, you can imagine actually having control over where a number of double helices line up next to each other, creating your structure. And the bottom one is just a reference to how we're going to represent double helices just as simple cylinders in our future pictures. So how does this work to build a larger structure? We've seen 2 double helices now be put together. How can we put a lot of them together how we want? Well, the method we've used has been pioneered by Ned Seaman at NYU, Paul Rotherman at Caltech, and here William Shih at Harvard, among others, and it's called scaffolded origami. And what it means is you take a large single strand of DNA. What we usually use is about a 7,000 base pair long piece of viral genome, and we add to it a bunch of what you see, the red strands there. And those are what we call oligostrands, the short strands I've talked about actually sitting down and ordering and adding to this long strand. And because we know that A pairs with T and C pairs with G, we actually can predict where those short strands will bind with the long strand. And that will pull the long strand together, and the red and black combination there is your double helix. And we'll see this happen with this animation. And they all basically come together with some heating and cooling into what we see here as a number of double helices all connected at various crossover points. And so we see a pleated sheet that actually we begin to feel how we can make a structure. So what if we connected maybe the 2 ends of the pleated sheet and we could wrap around and have a barrel? And that's where we got thinking at the beginning of this summer. So this is our structure. And basically it has 2 lids. Again, each of the cylinders represents a double helix. And this is a large barrel with a hole down the middle, and that's where, as we mentioned in the beginning, you might swap in whatever drug you're trying to deliver. And that would be just tethered by a piece of DNA, or you could think of other ways of potentially tethering it inside of the container. And the 2 lids keep it in there and keep the outside world out of there, which is really the important part. We see another view where we actually realize that there's a hole in the middle and that we can put our drug in there. And this container is about 30 nanometers cubed, so it's pretty much the biggest of its kind so far. And we think that that's actually going to be a great size to put proteins in, and antibodies, macromolecules, whatever you're looking to target. So what evidence do we have for actually building our structure? So we iterated through a number of designs, and this was a design we built in the end because when you try to take pictures of these structures under electron microscopy, they have to withstand enormous force. So we made doubly thick structures. As you saw in the picture before, each of the walls was 2 helices thick. And so that actually has allowed us to take negative stain electron microscopy images. You see one here, and it's on right about the right size frame because there can be a little bit of squishing, we think. And again, the sizes are actually just slight estimates because we're not sure exactly how tightly the DNA is binding. But the dark patch that you see in the middle of that structure where the 15.5 nanometer line is written is where we think stain is sticking to the middle. And then on the outside, we see where the DNA double helices are the 2 bands on either side. And so that actually is what we think a side view is, and the Illustrator file representing what we think is shown in that picture is right next to it. So more images. This is actually a different view of our structure. And the way negative staining actually works is that you basically drop your DNA down onto a grid and don't really have control over how it hits the grid and sticks. And so we think it's sticking in a number of different orientations, and we actually feel lucky as a result because we're getting to see different images. And so we think that these are top view images, and the stain is sticking in the middle. That's why there's a dark circle in the middle, and we see the top view of a number of these double helices. Again, these are pictures of just the barrel and not the lids. The lids would be in different pictures. And again, this is about the right size. You see our model of what we expect to see there, with each of the gray being these cylinders that we expect to see. So we're really excited about these images. But of course, we know that just taking pictures doesn't mean that the structure works how we think it will work. Remember, the goal from the beginning is protect something from the outside world. Protect that drug you're trying to deliver to a certain target. And that brings us to our protection assays. And this is sort of an ongoing process right now, but we have gotten some successful results, we feel. And the goal is to protect a payload. So what we've been working with is biotin and streptavidin because they like to bind tightly to each other, inside and the outside of our DNA nano boxes. And on the inside, we found that a streptavidin bead, or a magnetic bead covered in streptavidin, can't get to the biotin and can't bind with it. But the other picture shows that biotin on the outside actually does bind with the streptavidin beads, and we've been able to show this and reproduce it. So we're really excited about these results. But still, this is not the final picture. What we showed you with the design is that it has 2 lids. And here what we're relying on is the fact that the beads coated in streptavidin are too big to get to the inside of our structures. So that's why they're being kept out. And it's still significant, we think, because we are protecting sort of something on the inside world from the outside world. But we would like to see the lids attached to our structure and ultimately have lids that can attach and also detach so that we can capture something, close it with the lids, and then bring it somewhere and open it. And the bringing it somewhere is what you're about to hear about with cell surface targeting. How do we get these nanostructures to where we want to see them? So turning it over to Perry, we'll hear about cell surface targeting.
So in trying to get these nanostructures to cells, we actually explored two approaches that were more general in targeting. And the first one was these adaptamers, these molecular tethers. An aptamer is a single-stranded DNA sequence that can bind to a protein. And if you put two aptamers together by a linking region of complementary base pairing, you make an adaptamer, which was pioneered by the people at Tahirio Louis' lab. And we designed this adaptamer where you would have the thrombin binding domain on one side, the streptavidin binding domain on the other side, and linked by the complementary base pairing in the middle. And you can imagine switching out the streptavidin binding aptamer and putting another aptamer in there for a different protein, and thus linking thrombin with another protein. And if that protein were on a cell surface, you could therefore target thrombin to the surface of the cell. So why aptamers? As I just said, you can target substrates to the cell, but there are also other things you can do, like linking cells together with the tether and studying cell-cell interactions. Or you could bring two proteins together in close interaction and thus facilitate their interaction and make sort of a DNA enzyme. But that's for the future. For this summer, we created the streptavidin thrombin adaptamer and tested it using the beads that Katie just mentioned with the streptavidin on the surface. And then we added the adaptamer, which bound to the streptavidin, added the thrombin, to which the adaptamer bound as well, and then probed for the presence of this thrombin using anti-thrombin antibodies. And after bringing down the beads, we found fluorescence, and as opposed to the negative control, which did not have streptavidin. And this shows that the adaptamer is successfully linking the streptavidin to the thrombin. Another feature of adaptamers we explored was quenching. The linking region in the middle is dictated by complementary base pairs, and you can imagine adding a DNA displacement strand which preferentially binds for one of the aptamers, thus effectively knocking off the other aptamer, and then disrupting the adaptamer linkage and separating your two proteins. And repeating the fluorescence assay from before, we found that adding the displacement strand caused a reduced fluorescence in the beads that were brought down, similar to background levels. The second approach was expressing a protein on the cell surface to which you could bind any other thing. And this protein was streptavidin, which we've been using all along. And as Katie mentioned, it binds to the biotin molecule very strongly, and it's often used in labs to bind biotinylated nucleic acids or peptides. So the idea would be that if you had this on the surface, you could target any biotinylated DNA or protein to the surface of the cell. And we put this on the surface using the Lpp-OmpA display vehicle by these guys up in the upper right. And this is a fusion protein made up of the LPP, or lipoprotein signal peptide, fused with transmembrane domains from OmpA, or outer membrane protein A, and then finally the surface protein. And they were able to express proteins on the surface such as antigen binding domains or beta-lactamase. And using biobricks, you can actually fuse these protein domains. Standard biobricks have the spacer nucleotide in between the X1 sites, the SPE1 sites, and the part in between. If you remove this spacer nucleotide, you can actually have a mixed site, which is 6 base pairs long, and therefore reading frame is maintained in between the protein domains. And you can imagine you can use this anywhere, putting together protein domains, which is what they do at Ira Phillips and Pam Silver's lab. And so this would be the construct with your promoter and ribosome binding site, and then your fusion protein of the LPP signal peptide, the OmpA transmembrane domains, and your streptavidin. And you could also switch out streptavidin for other proteins that you would like to express on your cell surface. And so in trying to test for expression of these, we did a couple of Western blots, and it showed inducible expression. The histidine, that's 6 histidine amino acids at the end of the construct, and so they're being detected by the antihistamine antibody, and because they're at the end of the construct, it shows that the whole protein, the fusion protein, is being expressed in frame. And also, because the anti-streptavidin antibody is binding, the streptavidin domain is folding in such a way such that the streptavidin antibody can bind. And so this is just expression of it, and for the future, we'll be trying to add a biotinylated oligonucleotide, which also has a fluorescent tag on it to show binding to the membrane. And in the future, we also hope to put this with nanostructures and try to target nanostructures to the surface of the cell. And now for our second project.
So the second project Harvard iGEM pursued this summer was reconstituting a cyanobacteria oscillator into E. coli. So cyanobacteria are photosynthetic bacteria which possess a circadian rhythm. Now, they've evolved this rhythm over billions of years. This rhythm, as an oscillator, could be used to potentially do useful work. However, the model organism of the time is E. coli. So what we wanted to do was move the oscillator from cyanobacteria to E. coli. Now, the applications of a biooscillator are multifold. Imagine this is the output of an oscillator of time. The obvious one is a clock, where the amplitude of the signal determines the time of day. Now, right alongside is a nightlight, which, for example, can be a luciferase reporter. Other applications, though, can include time drug delivery. For example, you could release melatonin at night to aid in sleep. Also, during drug synthesis, many times reagents need to be added at a certain time of day. So this can be automated under a biooscillator. Of course, in biocircuitry, you can use this with biobrick systems. And research in biooscillators can be used to investigate the natural circadian systems. So many of you know the RIF oscillator. It was developed by Elowitz at Caltech in 2000, and it's a system of 3 proteins that suppress each other and create an oscillation. Now, this was a breakthrough in synthetic biology, and for good reason too. However, if you look at the graph of fluorescence over time, you see that after 10 hours, it becomes unstable. Now, in contrast, our biooscillator from cyanobacteria, it's also 3 proteins. However, it's been shown to be stable over the lifetime of the organism. This is only 24 hours of data. But the oscillator is robust, and it's temperature compensated, meaning that changes in temperature will not change the period, which is still at 24. So as you can see, there's much potential here for an oscillator. So to go over the biochemical mechanism of the Chi clock, again, it's 3 proteins, Chi A, Chi B, and Chi C. And the phosphorylation state of Chi C is the output of the system. Now, bear in mind that this is a bit simplified in that in the actual system, this would represent the average value of Chi C phosphorylation. So Chi C can autophosphorylate and dephosphorylate, but when kept stable, or when kept alone, it will be in a dephosphorylated state, or prefer a dephosphorylated state. Chi A promotes the phosphorylation of Chi C, while Chi B inhibits Chi A. So if you put this model together, you can see that, again, the output varies with phosphorylation state of Chi C. Now, an important fact here is that the system is transcriptionally translation independent. This was verified by the Kondo lab in 2005. In other words, these 3 proteins were put in a plate, and they were shown to oscillate for 5 days in vitro. So this means that we shouldn't have potential problems migrating the system into E. coli, because we won't have to move transcription factors and such. Also, the period can vary from 14 hours to 60 hours based on point mutation. So there's much flexibility for a potential for an oscillator. So again, our goal this summer was to reconstitute the cyanobacteria Chi oscillator in E. coli. We've managed to create Chi A, Chi B, and Chi C biobricks, put them under promoters to form functional biobricks, and we've expressed these proteins in E. coli, and we've verified interaction between them, which is a large step towards oscillation.
So I'm here to talk in more detail about our results, and I'll start with the constructs we created. So we've added the parts you see here to the registry, and we've created biobricks of the Chi genes Chi A, Chi B, Chi C. We've ligated them with like promoters from the registry, and we've also combined them with each other to make Chi A plus C and Chi B plus C parts with promoters. So our most exciting result is that we've demonstrated that at least 2 of our proteins, Chi A and Chi C, are actually being expressed and interacting in E. coli. The experiment we did to demonstrate this, we started by transforming 3 of our constructs into E. coli. We grew the cultures, we sampled them, we lysed them. We formed a Western blot with anti-Chi C antibodies. And the result is in the image you see here. The 3 lanes are from left to right, sorry. Yeah. The 3 lanes are from left to right, Chi C, Chi B plus C, and Chi A plus C construct bearing samples. We believe that the top band in each lane is phosphorylated Chi C, The bottom band in each lane is non-phosphorylated Chi-C, which runs slightly faster. So how do we interpret these results? Well, let's take a look at the first lane. You'll notice that when Chi-C is expressed by itself, there's a small amount of phosphorylated Chi-C, but most of the Chi-C is non-phosphorylated. So you will recall this is consistent with our model, which says that Chi-C will spontaneously phosphorylate but prefer the dephosphorylated state. When you add Chi-A, however, you expect to see a higher level of phosphorylated Chi-C. And that's what you see here. When in the lane 3, you can see that the top band is much darker relative to the second band, and also relative to the first lane. So this confirms our prediction that the Chi-A and C molecules are interacting as they should. Finally, in the middle lane, you see what happened when we add a Chi-B and Chi-C alone. And our model predicts that this lane should be more or less identical to the first lane, because Chi-B's effect is to inhibit Chi-A. So if Chi-A is gone, then Chi-B should have no effect on Chi-C. And you notice that, in fact, it appears the ratio of phosphorylated Chi-C to non-phosphorylated is about the same as in lane 1. So we concluded from this experiment that our Chi-A and Chi-C are being expressed in E. coli, and they're interacting. We don't have positive proof that Chi-B is interacting yet. That has to wait until we make a Chi-ABC construct. But at least the results in lane 2 are consistent with our predictions. So the next step is to actually verify that our oscillators are oscillating in E. coli. There are a couple of complications that make this a bit tricky. Here's a model of our oscillator again. You can think of each complex of the Chi-A, B, C molecules as a single clock. So our problem is that all these clocks are independent. If you have a bunch of these clocks floating around, there's nothing to ensure that their phases will agree. This is a problem for us. We actually need our cells to be synchronized, because when we take a sample of many cells, if these cells are out of phase, we won't be able to detect an oscillation over time. We also need a way of synchronizing the Chi clocks within a cell. The problem here is that as you translate more Chi proteins, the new clocks that are produced will be out of phase with the existing ones. So as time goes on, you expect to see the oscillation die out. And we made a computer model of this effect, and you can see the graph here, that the oscillation in Chi-C starts strong and then dies out as more and more Chi-C is produced. So how do we solve these problems? Well, we think we have a solution that will solve both of these. And that solution is to use pulsed expression of the Chi genes. That is, we'll induce expression of the Chi genes for all the cells in a culture at the same time, then stop expression shortly after. So this solves both the problems of synchronization, because for between cells, we're starting all the clocks at the same time, and within a cell, we're not producing clocks for very long. So we designed some experiments using this synchronization solution, and we hope to have those running very soon. In conclusion, Harvard iGEM pursued 2 projects this year, drug delivery and biooscillation. You've seen DNA nanostructures capable of carrying and protecting molecules. You've seen 2 approaches for targeting the cell surface, and you've seen the reconstitution of the cyanobacterial oscillator in E. coli. None of this would have been possible without the help from the people on this slide. We'd also like to thank MIT for hosting the Jamboree, and you, the audience, for listening. Thank you very much. So, yeah, now if you have question marks, we have periods. So.
Yes.
The naturally occurring Chi system, it must have some external interface that keeps it in sync.
Yeah, so cyanobacteria have light-sensitive mechanisms. They're photosynthetic, and so they're reset every day by the night to day transition.
So if I was using your biobrick, I would also want a hook that I could just plug into.
So yeah, ideally you'd have some way to actually synchronize the clock or to reset the clock more accurately. We don't have a mechanism for that in E. coli yet. Obviously, E. coli themselves don't have light-sensitive organs, but that's how you do.
Streptavidin itself is a pretty tenacious tetramer. What's being expressed on your membrane? Do you have a monomeric unit or
Right.
several membrane proteins together to determine the tetramerize?
That's a great question. Yeah, streptavidin is a tetramer, and so the question was about streptavidin being a tetramer and also the affinity of streptavidin and biotin. Streptavidin and biotin, it's huge, like the binding. The dissociation constant is something along the order of 10 to the negative 15. And so there's actually been research, such as in the Ting lab at MIT, like making monomeric streptavidin, which would actually bind by its in less strongly, more along the lines of 10 to the -7, 10 to the -8, and so you can have reversible binding. And so theoretically, we would only have a monomeric streptavidin on the surface. I'm also using, from Felice Aslan's lab at BU, a single-chain dimer streptavidin, which would potentially be a better binder, but not quite as strong as tetrameric streptavidin. And if we did want to have tetrameric streptavidin, one possible direction is to create a longer tether between the membrane display vehicle and the streptavidin so that there is some sort of three-dimensional flexibility to form a tetramer at the surface.
So the molecular, you want to answer?
Okay, well, yeah, so from the, right, so the question was for clarification on the mechanism of the oscillation. If I go back to the previous slide here, pretty much it's expressed here where the phosphorylation state of Chi-C, you see as it changes, when it's fully phosphorylated, then the oscillation level is high, so there's high expression. But when it's at a low phosphorylation state, then the oscillation level is at the lowest part of expression, pretty much. Oh,
so what happens is, so you have these three proteins. Chi-C creates a hexamer with itself, or a hexamer, I should say. Chi-C creates a hexamer and will phosphorylate in that state, but in the phosphorylated hexamer is not very stable unless Chi is also present to hold it there. It's not, the exact molecular mechanism is not known yet. It is known that this general model holds. So, hope you answered that question.
So the question is the complexity of the DNA all coming together, how often does that not work out, and how often do you have structures that don't look like you expect? So I guess to answer honestly, it's hard to say how often that's happening exactly, because when they don't form correctly, you pretty much can't image them because they're not going to hold up to the stain and stay a solid block. And on the electron microscopy, taking pictures of them at least, it is a difficult process that the imaging itself doesn't always work. And so you do see a lot of junk a lot of the time. And so potentially that's some that aren't folding. What we think is really that's more an artifact of the staining process. In terms of the complexity of the folding up together, it actually is, we think, highly predictable. A pairs with T and C pairs with G pretty much is always happening. So as long as you're not really making mistakes in design, and we spend a lot, probably the whole first half of our summer was simply in design and laying out letter by letter exactly what each nucleotide was going to be. I guess if you don't make those mistakes, hopefully the structures are folding relatively reliably. It's a very simple structure process after you order the DNA. You really are just following a very basic protocol that only takes a few hours to fold them up. So we think that they fold with relative reliability.
Repeat the question.
Right. So the question is, because there's no reporter method, how exactly are we going to get a method of reporting into the oscillator? So this is true, but there's a couple proteins that we know. If you've seen the Chiba this year, is also working on the cyanobacteria oscillator, and we don't know yet the link between the transcription factors in cyanobacteria and the Chi oscillator system. That's yet to be derived. But hopefully, first off, this research here will allow us to port piece by piece into cyanobacteria or into E. coli to actually figure out what's missing there. And once that happens, then you have the output. The second thing is right now we know there's SAS-A and SAS-A and RAPA-A, I believe, in cyanobacteria, which are theorized to be part of the output system. So there are homologs in E. coli, which you could then potentially take advantage of to have output. in E. coli. So.